Other factors include larger exposure. So there's a larger surface area to volume ratio in the small body, and there's a higher respiratory rate, so potentially greater delivery of toxicants. There's slower hepatic metabolism of xenobiotics, and as I just pointed out, there's altered cellular extrusion of some of these compounds. I also want to introduce the idea of immunologic priming. Microglia and immune cells are resident in us, and they're highly sensitive to factors that occur during development, and these seem to affect their long-term behavior. And we'll talk about that in a few minutes. And there's also the issue of microbiome development that occurs very early in life and affects the immunologic responsiveness of the organism for the rest of its life. I want to talk briefly here about genomic priming or epigenomic modification. People have probably heard about epigenetics. So epigenetics are modifications to the DNA sequence that are not directly part of the DNA sequence. So it's usually methylation or acetylation of DNA, and it affects the way it is expressed later in life. So these mice here are actually genetically identical. The mouse on the left, its mother received a standard diet during pregnancy. The mouse on the right, its mother received a diet rich in folic acid, which increased the methylation of its genome. And increased methylation is usually related to decreased gene transcription. So with this decreased transcription, this mouse is relatively protected from the obesity, diabetes, and cancer that this mouse on the left is at higher risk of.